we have a fair number of questions, and so we're going to try to get to some of them. And some of them are more specific, some are more larger context, so I'll try to include a mix. First one is a little bit specific, and it's directed at Dr. Barber, which is there a utility of in silico models in risk assessments of mixtures?
Yes, there is. I think there's a bit more work to do. So if you think about a single chemical, it can interact at many different parts in a biological system, hit different receptors. And that thinking is exactly the same thinking that would have to be applied to a mixture of chemicals. So rather than thinking of one chemical that might hit several different important toxicity endpoints, you can consider a mixture of chemicals which does the same. So the same thinking needs to be applied. And of course, the complexity is validating that with test data sets, because you need to characterize that mixture as well. But the concept is exactly the same.
Great. A couple of questions about the PBPK modeling, and so I'll just try to combine them. So for Dr. Ellison and Dr. Dent, first of all, have you found that there are specific phys-chem features of structures that stood out as having more or less reliable predictivity for risk? And so can you, I guess, connect phys-chem characteristics to risk using the PBK models? And then also, are you able to track concentrations of individual metabolites over time using these types of models?
Corey, do you want to have a first stab, or would you like me to?
I'll answer the second one, and then for the first one, I think that ties a little bit more to the toxicodynamic piece. I'll let you tackle that one. So as far as tracking metabolites within these kinetic models, yes, that can be done. It requires knowing similar information about the chemical. So knowing some things such as, excuse me, such as the phys-chem properties, and then also clearance rates. So ideally, you would take that metabolite and have a stable form of it and generate some of that data in vitro, and then it just becomes kind of a sub-model in your kinetic modeling. So the parent model would just link to the metabolite.
Thanks, Corey. So in terms of where we have problematic substances for PBK modeling, I guess anything which is tricky to test in vitro would be problematic because of the in vitro inputs. So anything that's particularly volatile, or where we have problems with solubility. But of course, also there's the issue of specific transporters that might be involved with the transport of the substance. If we don't have any in vivo experience of that, it's with the existing tools quite difficult to have confidence in a PBK model where you have specific transporters that are involved, unless you take them into account in the model.
Okay. Interesting question about applying this type of risk assessment to other sectors. Can you think about next generation risk assessment for pharmaceuticals where there might be 2 points of departure? So one for adverse effects, but you're also looking for efficacy. I know that's a little maybe sort of beyond the topic, but it's sort of interesting to think about.
Yeah, I mean, with pharmaceuticals, where you've got a drug which is systemically available and intended to do something, then clearly the type of lower tier NGRA that I described with the protection not prediction is going to be very problematic, because you will, by that very nature, have bioactivity at relevant exposures. So I would see that as needing to tip into much more of an adverse outcome pathway based, much more mechanistic approach, which I, to be honest, don't necessarily see in the foreseeable future. That's little bit of a long way off, and maybe along the lines of Chris's timescale that he was showing earlier.
Mm-hmm.
And I certainly think some elements of these can be used within the process of pharmaceuticals. So modeling, kinetic modeling certainly is part of, I think, the pharmaceutical pipeline. And then even some of the toxicodynamic assays could be used as screeners, potentially to weed out prototype drugs. But yeah, for the ultimate risk assessment, it's probably going to be a little bit harder.
Okay, great. A couple of questions for Dr. Walker. Wondering about the metabolic activity of 3D skin. Well, maybe this is for more than Dr. Walker, but I guess respond in terms of the models that we've all presented, skin models or other models. So the metabolic activity of those three-dimensional models and what we know about them. And are the data from these cells mostly primary cells, or are they cell lines?
So I guess I presented on liver models rather than skin models. So I'm guessing the question doesn't quite relate to me. But in terms of the liver models that I presented, then certainly CYP expression that we see on metabolism activity is quite comparable between the HepRG cell line and human liver and human hepatocytes. With regards to human hepatocyte models, it's very donor specific. What we typically see is comparable CYP expression into the individual donor. So if, for example, if it has low CYP3A4 as a donor, then it will have comparably lower 3A4 expression in 3D to other more stronger expressing CYP donors. Whereas HepRG tends to sit somewhere, I would say, at the higher level of the CYP expression, or majority of the donors.
Okay.
I don't know if anybody else wants to comment on skin 3D. Obviously, we have skin 3D models, but I don't know whether
right. Okay, that's okay. We'll move on. Another question for you, actually. Do you expect that different cell types can better represent relative risk to target tissues or off-target tissues more accurately than HepG2? So,
yes,
right.
absolutely. I mean, HepG2 is used as just kind of a surrogate cell type. I wouldn't even say it's specifically used for the liver due to the low metabolic activity that we see. It tends to be quite a responsive cell type. You know, it's quite sensitive in general to in comparison to a lot of different cell types. So I guess in terms of risk assessment, it's quite useful if you want to take a conservative approach. But certainly when you're talking about specific organ models, then it's really important to consider an appropriate in vitro model or organoid. Obviously, things like cardiac cells, then there are lots of great leaps forward using iPS cells and recapitulating the function of a cardiac cell. And obviously, with regards to liver, then metabolism is a key element, as well as uptake and export of drugs within different kind of export pathways, or into biocaniculi, for example, expression of different transporters.
Right. Okay. We have another question. It's directed at Dr. Barber, but I think it could be for most of you to think about or to jump in on. So we're talking about systemic endpoints, you know, the chronic systemic endpoints like target organ toxicity, developmental and reproductive toxicity, and asking for comments about whether we have sufficient knowledge to start applying AOPs for those endpoints. Are we still needing to build that knowledge, and how can we, do that to take advantage of this type of strategy for those types of endpoints?
Would you like me to have a go at that, Christy?
Sure, yeah, that'd be, go ahead.
So I think it depends on the type of safety assessment that you're doing. But for me, if we're trying to operate in a protective, not predictive mode, this is more about looking at the different measures of in vitro bioactivity that we've got and seeing if the points of departure are protective for those effects that you just mentioned.
Yeah.
So are they protective for effects on development, even though they might not predict that something would cause malformations? If the point of departure is giving you that level of protection, then do you really need to know that at very high exposures in animals it would cause developmental toxicity? That's the way that I would tackle that. I don't know if you think differently, Chris.
Yeah, sorry, I was struggling with my unmuting my technical inability. Yes, so in order to be comprehensive in the toxicity pathways, in order to make a safe decision about a systemic or an organ endpoint, is really challenging. You're back into the Donald Rumsfeld's known unknowns and unknown unknowns. But the way I'm looking at it really is we've spent quite a lot of time over the last few years looking at developmental toxicity. And we believe we have captured the toxicity pathways for all of those known chemicals that cause that apical toxicity. So you then get to the point where there may be something you don't know, which is unique and novel, but it's probably still more accurate than hoping that the animal model you're using is going to pick up the same toxicity that you might see in a range of in silico models. So it is really challenging, and the prerequisite really is an understanding of all of the known pathways that lead to that toxicity, and a good coverage of chemicals which have shown that toxicity. If those two align up well, then the risk of discovering a new pathway to toxicity for that endpoint that hasn't been seen with 100-odd chemicals before is pretty low. So it's never going to be perfect, but it's probably going to be pretty close. We're not quite there yet, though.
Yeah. Okay, great. And I have to say, I think that's probably a really good place to end the conversation. We still have some questions we haven't gotten to. We'll try to get to those after the webinar if we can send some messages. I really appreciate all of the presenters for sticking around to answer some questions, and for all of you for listening. And I'm really inspired by all these presentations and by the sort of organizational work that the AFSA coalition is doing to try to move this next generation risk assessment forward. So great work, everybody. Thanks for your presentations, and have a wonderful rest of your week.